Hello, and thank you for watching our presentation on the development and applications of cell-derived extracellular matrices. I'm Travis Block, Principal Scientist at STEMbiosis. And at STEMbiosis, our core technology is the development of cell-derived extracellular matrices to make cell culture more biologically relevant. Now, I always like to begin my presentations with a little history lesson. So Alexis Carrel actually won the Nobel Prize for developing vascular sutures and enabling organ transplants, and Montrose Burroughs was a student of his. And while doing microscopy studies of capillary bed structures, the two of them actually discovered modern cell culture. So they were taking these thin slices of tissue and putting them on microscope slides, and they observed cells migrating out of the tissue onto the slide. And so in they published a paper where they described the new methods for cell culture of mammals. And interestingly, cell culture hasn't really changed a whole lot since then. In traditional cell culture, we tend to dissect a tissue, then that tissue sample is either enzymatically digested or mechanically disrupted in order to isolate the cells of interest, and then those cells are put into tissue culture conditions. And those conditions are typically very simple. Often it's just tissue culture polystyrene. Potentially the plastic dish is coated in extracellular matrix proteins like fibronectin, collagen, laminin, or some mixture of several extracellular matrix components. However, really very little is done to recapitulate the natural environment. And there's something wrong with this system. The problem is that when you really stop and think about the environment these cells are coming from, the cells are likely to respond just as strongly to the lack of signals that they should receive as to any signals that we do give them. And so oftentimes when we are trying to study experimental conditions, we're really studying the response of cells to the bizarre environment we're placing them into. So when we talk about cell culture, there's really three key components that we need to keep in mind. So there's the substrate or the extracellular matrix, there are the cells, and then there's the soluble factors. The soluble factors get a lot of attention because they're very easy to manipulate in culture, very easy to do iterative testing on, very easy to do dose response studies. And the cells get a lot of attention because often they're the dependent variable in our studies. So we manipulate soluble factors and then we assay cell responses. So the two of these get a lot of attention, whereas the extracellular matrix frequently gets ignored. And there may be several reasons for this. One of them is that this is really very difficult to assay, partially because the ECM is influencing cells in a wide variety of ways. So there's structural cues, there are physical cues like the mechanics, the stiffness of the substrate, things like surface energy can play a role, and then there's also biochemical effects. So different binding sites may activate different ligands on cells and cue different intracellular signaling cascades. And it's very difficult to modify one of these signaling factors without modifying all of them. So if you try to take a component out of the extracellular matrix to change the biochemistry of it, you may also be changing the structure, you may also be changing the mechanics. And so it's very difficult to isolate variables when studying the extracellular matrix. However, it really shouldn't be ignored. So this is an image of a cell in its extracellular matrix, in its native niche. And we're taught from a very young age that cells are the basic building blocks of living organisms. However, when you think about a human, if in theory you could isolate every single cell from a human and spin them all down in a giant centrifuge, they really wouldn't take up all that much space. And that's because even when we talk about the dry weight of humans, it's mostly extracellular components. We are organisms that are composed of extracellular matrix with some cells spread throughout it. And in this image, it's very striking, and you can see lots of communication going on between the matrix and the cell. However, when we take a cross-section of the cell, the image becomes even more. So until I label this image, it's actually very difficult to tell where the cell ends and the extracellular matrix begins. And that's because the extracellular matrix is interacting directly with cytoskeletal components. So here, in the bottom left-hand side, we have the cell, and in the upper right, we have the extracellular matrix with a phospholipid bilayer going across the image. Here we can see that there's constant communication between the ECM and the cell that just cannot be discounted in cell culture conditions. And over the last 15 or 20 years, we've seen more attention being paid to the role of the microenvironment in regulating cell fate. However, it's mostly been with very rudimentary models. On this slide, my favorite example of this is when we look at this PNAS paper from 2010 in the middle of the screen. In this study, all they did was make little cookie cutter cutouts on tissue culture plastic plates. So the biochemistry and the mechanics of the substrate were exactly the same in both groups, or in all the groups, but they forced cells to assume different morphologies by cutting out shapes in the substrate. And just by forcing cells to assume a different morphology, you could change the way they sense the substrate, and it impacted gene expression and differentiation. And so this is an excellent example of why it is so difficult to study the extracellular matrix. Because here, even though the substrate was actually the same, changing the structure impacted how the cells sensed the other signals, and it changed gene expression and differentiation. And so because of this, this shows how difficult it is to recreate the matrix artificially. And so our stance has been that we want to make this as natural as possible. And so even though cell culture conditions haven't evolved much over really since 1910 when it was first discovered, we want to create the next evolution of cell culture that's going to be more biologically relevant. So we want to take cells out of a native tissue and put them right back into culture conditions that resemble a native tissue so that the cells can behave as they would in the body. And so the way we do this is we take stem cells, we seed them in a dish, we culture them to confluence, we induce them to secrete a matrix, and then we gently remove the cells using a non-ionizing detergent. The detergent is washed away, and we're left with the matrix the cells secreted in the dish, and we, as much as possible, we leave the biochemistry and the structure and the mechanics intact. That matrix is then dried onto the bottom of these tissue culture dishes, so the end user just has to add PBS or media to rehydrate the matrix, and then they're left with the substrate that the cells created at the bottom of the dish. So to give you a better idea of what I'm talking about, this is a very up-close look at the matrix. You can actually visualize it in bright field microscopy. But the fact that you can see it in bright field doesn't interfere with your ability to also visualize your cells in bright field. And you can do fluorescent imaging on the matrix. So on the left panel here, we have actually matrix components being imaged. So this is collagen 6 staining. And on the right, we have cells stained fluorescently on our matrix. Now, where this gets really interesting is when we start to get into the applications of the matrix. So these next few slides are going to deal with mesenchymal stem cells, because that was what a lot of the early data was produced using. And in general, when isolating primary cells, oftentimes very high concentrations of serum is used. And a big reason for this is that primary cells aren't naturally very adherent to plastic in many conditions. And so we use serum so that it will coat the dish and make it stickier and stick on the cells and make them stickier so that you can get them to stick to the substrate. And there's several products that are commercially available that advertise the ability to isolate cells in xeno-free conditions. However, in our experience, those products have not worked well for us in isolating primary cells. However, with our CELLVO xeno-free matrix, we're able to efficiently isolate primary cells in totally xeno-free conditions. They rapidly adhere to the matrix and begin to proliferate, so that we can get very high yields of primary cells that are xeno-free. But I never really am too concerned about the cell number because it doesn't matter how many cells you can get if they're not high quality. And in this image, I know you can't see the labels on the left, but what I want to focus on is that the gene expression rapidly changes when you put these cells onto tissue culture plastic. And so what happens is as soon as you take cells from the body and put them onto a simple substrate, they begin to upregulate the matrix components they're not seeing. For instance, if you put an MSC onto a plastic dish, they're likely going to start upregulating things like fibronectin and collagen 1 and collagen 12 and collagen 6 that they expect to see in their matrix that they're not seeing in their tissue culture environment. And the reason this is so critical is that gene expression changes don't happen in a vacuum. There's crosstalk between just about every gene expression pathway. So we can't expect a cell responding to its microenvironment and upregulating these matricellular proteins to not impact other gene expression pathways. Another really interesting piece of data on our matrix is shown in this graph. So across the x-axis, we have different donors of bone marrow MSCs. On the y-axis, we have cell doubling time. And so these are cells that are plated onto either tissue culture plastic or CELLVO Matrix. Universally, the cells grew faster on our CELLVO Matrix. But again, I'm not too concerned with the proliferation rate of cells because in practice, you could always just grow your cells for another day if you needed more. But what I'm very interested in here is the consistency of different donors on the CELLVO Matrix and the relative inconsistency of donors on the tissue culture plastic. Now, this piece of data fascinates me because I know that there's this really interesting source of bias in both academic and industry research where we tend to only study cells that behave bizarrely well in the culture conditions that we create for the cells. So if you thaw a vial of cells and they're not growing well, you're likely just not going to thaw those cells from those donors or from that donor again, and they'll sit in a cryopreservation tank for 5 more years. Somebody else comes along and thaws those cells. They still don't do what they want them to do. They sit again until eventually somebody tosses them out. And so we believe that by putting cells in a more natural environment, we're able to get more consistent responses that enables us to do our work, do our studies on a more biologically representative sample of donors, rather than selecting for donors that behave bizarrely well in odd tissue culture conditions. And so even though the data I just showed you was from MSCs, we've had in our own hands and in the hands of our collaborators and customers, a variety of different cell types have been used on CELLVO Matrix. And so in general, we recommend CELLVO Matrix for use with mammalian adherent cells. So in summary, CELLVO Matrix creates a natural protein-based environment for isolating and expanding adherent cells. It works with a variety of cell types, and in general, what we see is it improves yields of primary cells, it improves viability, and it improves phenotype retention. Cells that are grown on CELLVO Matrix are more responsive to growth factors. And what we mean when we say this is a very interesting paradox. And so what we observe is that while phenotype retention is better on CELLVO Matrix, the cells are simultaneously more responsive to induced differentiation. And it's very difficult to parse out mechanistic interactions when we're talking about the matrix for the reasons I discussed earlier. But our hypothesis here is that because cells aren't busy expressing all the matricellular proteins that they're not getting from their environment, there's not a bunch of signal noise in their gene expression, and so their cells are more capable of being responsive to experimental variables. CELLVO cells and matrices fit into existing workflows, and so you can still use your same enzymes to attach cells, you can still use your same media. We're just creating more biologically relevant microenvironments for your culture. And cells have a more native phenotype when cultured in CELLVO Matrix. So hopefully we can help you to do work that's going to be more translatable. So in short, we enable more biologically relevant cell culture. Now I'm going to move on from talking more generally about our technology to talking about a specific application of cell-derived extracellular matrices that I believe is an excellent case study for the importance of biologically relevant culture conditions. So this is work done with Joachim Cohn's group at the New Jersey Center for Biomaterials, working on the growth of human articular chondrocytes. So this is a particularly interesting problem because in culture, chondrocytes rapidly de-differentiate towards fibrocartilage, impairing their ability to form cartilage in vivo. And so what happens is we isolate chondrocytes, and they have very rounded stellate morphology, and very quickly in culture they become very fibroblastic, and they lose their collagen 2 expression, and their collagen 1 expression shoots up. And so typically when people are doing research on chondrocytes in culture, they're really working with cells that have very little resemblance to the cells that are found in the body. And so we began by producing extracellular matrices, either from bone marrow mesenchymal stem cells or from articular chondrocytes. I'm not going to focus too much on any of the specifics on this slide or the next one, but what I want you to see is that cells produced from different tissues retain tissue-specific properties and secrete a matrix that has unique tissue-specific properties. And so cells from either articular cartilage or from bone marrow produce cells with distinct topography, elasticity, and surface, as well as distinct biochemistry. And many of these differences are predictable based on the tissue source. Then when we put human articular chondrocytes onto the matrices, the bone marrow MSC-derived matrix, the CELLVO matrix, or the articular chondrocyte-derived ECM, we see that either of the matrices supports rapid cell proliferation, so much so that we're able to get 400 to 550 fold expansion in about 2 weeks on the matrices, whereas on TCP it takes nearly 4 weeks to get a similar amount of expansion. Moreover, while we're expanding those cells, despite growing much faster, the cells cultured on the articular chondrocyte ECM retain their native morphology, this rounded stellate morphology that's typical of adult mature chondrocytes. Whereas on tissue culture plastic, the current clinical standard, the cells become very fibroblastic. Not only did they retain the morphology, but cells cultured on the tissue-specific matrix maintain a much higher collagen 2 to collagen 1 ratio, a key marker of chondrogenic potential. Then those cells pre-expanded on the matrix are capable of secreting a matrix that's very cartilage-like in a micromass culture, such that it is rich in collagen 2 in the green here, and rich in glycosaminoglycans, which is key to cartilage mechanics. And so only the cells grown in the tissue culture, in the tissue-specific extracellular matrix conditions, retain the ability to secrete a cartilage-like matrix after culture. So in short, cells from bone marrow and articular cartilage produce tissue-specific matrices, and culturing articular chondrocytes on a tissue-specific extracellular matrix results in cells that maintain chondrogenic potential in vitro and maintain the properties of mature chondrocytes. And so this is an excellent case study for why it's important to use a tissue-specific matrix for your research, but it's also an excellent case study for why we sell our cells. So CELLVO cells are always sold from healthy tissues, from young donors, and they've never seen artificial substrates. And we do this so that we can provide researchers with cells that resemble those found in the body. So with our chondrocytes, because we sell them either at P0 or at P1 expanded on a CELLVO matrix, we're able to sell cells that still have a very high collagen 2 to collagen 1 ratio that still look like chondrocytes from the body, whereas competitors may be selling cells that no longer resemble any cell type found in the body. So if you're interested in seeing more detail on this work, it was recently published in Active Biomaterialia. And in summary, the ECM produced by either bone marrow MSCs or articular chondrocytes had tissue-specific structure, composition, and function. And when we control for population doubling level, cells cultured in the tissue-specific matrix maintained gene expression morphology typical of mature chondrocytes. And they were then able to make a more cartilage-like matrix in pellet culture following expansion. So, articular cartilage-derived ECM may represent an ideal substrate for the expansion of chondrocytes. I'd like to acknowledge the people that did the majority of the work for the data that was shown here. Young Mao and Anya Singh Varma at the Rutgers, New Jersey Center for Biomaterials, and Ann Sheldrake and Rachel Leaf at Stembiosis. Here we have the products currently being marketed by Stembiosis under the CELLVO brand. We have our original CELLVO Matrix and the CELLVO Xeno-free Matrix. We have several sources of mesenchymal stem cells that are all isolated in totally xeno-free conditions. We have cord blood endothelial progenitor cells and human chondrocytes that are isolated on our CELLVO Matrix. And we recently launched our first media products. You can expect our CELLVO Chondro Matrix to be launching in the fourth quarter of 2019, as well as our CELLVO Matrix Plus, a new matrix produced from perinatal stem cells for the expansion and differentiation of pluripotent stem cells. So CELLVO Matrix Plus is going to be an intact matrix produced by human cells to provide structural, mechanical, and biochemical cues necessary for biologically relevant cell culture. It's comparable in efficacy to competitive technologies for self-renewal of pluripotent cells, but it's much easier to use. Rather than having coating steps and having to cool tips so that you're not gelling a substrate, all the end user has to do is add PBS or media to rehydrate the matrix prior to use. And aside from that, it will fit into your existing workflows. And what's been very interesting is that we have accumulating evidence that while it's comparable to competitive technologies for self-renewal of pluripotent stem cells, CELLVO Matrix Plus actually supports more mature phenotypes of differentiated progeny from stem cells. So this is a really interesting application of this technology because the inability to get mature phenotypes from differentiated cells has been a problem plaguing the field, and we think we have a really interesting solution to that. So we have more data coming on that soon, and we're excited to share that. With that, I'd like to thank Caltag Med Systems, our distributor in the UK and Europe. And if you have any questions, you can reach out to Caltag Med Systems, or you can reach out to Stembiosis directly through www.stembiosis.com or info@stembiosis.com. Thank you.